So what we're trying to do at Bone Therapeutics is really try to reshape the orthopedic landscape and reshape the way patients are treated in orthopedics. We dream of a world where our game-changing bone therapies will help rebuild literally people's lives. And how do we do that? So we're located in Belgium. We are founded in 2006 as a spin-off of the University of Brussels. We currently have two products in our pipeline. One cell therapy product called ALLOB. It's an allogenic cell therapy product that I'll cover in more details later, meant to regenerate bone, reshape bone, which makes a lot of sense since the skeleton is a naturally regenerating system, and helping this system in some cases prove to be useful. We are currently in two indications with that product ALLOB. One of them is entering phase 3 before the end of the year. The second one is a non-cellular product. It's a viscosupplement for osteoarthritis of the knee, an improved viscosupplement that I will also cover. So we're really trying to focus on those markets that are currently underserved and large. Most of the innovation, or a lot of the innovation, is coming from medtech companies, but this innovation comes in very small increments, and we're trying to propose something different. As a company, there are a bit less than 100 people working in our company. We have our own GMP manufacturing facility, which allows us to totally control the production of our cell therapy product. And finally, we're listed on Euronext Brussels and Paris since 2015. So if we have a look at our pipeline, so as I said, two products, ALLOB in red, JTA004, the viscosupplement in green. Those two products, the first product, ALLOB, the allogenic product, is currently studied in two different indications. The first one is lumbar spinal fusion. It's a surgical procedure, very common surgical procedure that takes place when the intervertebral disc is diseased and needs to be removed. At that time, the two vertebrae need to be fused, so they are immobilized and the pain disappears. Unfortunately, in many cases, in about 30% of the cases actually, the patients are not satisfied with their surgery because the fusion didn't take place correctly. So what we do is we act as an add-on to that surgery. We place our bone-forming cells in the operation so the fusion takes place in a better way. It's a very large indication. If you're only looking at the US and Europe, there are more than 500,000 of those procedures taking place every year, and it's a growing market. We completed a phase 2A, and we will unveil the full results in the summer of this year. That's for the first indication, lumbar spinal fusion. The second indication is called delayed union fracture. So for that one, we will cover that in a bit more details later. It's also a very large indication. We think that there are about 700,000 cases in the large Western markets. This is an indication where we treat severe fractures, so fractures requiring osteosynthesis material like plates, screws, pins. Typically, such a fracture would heal in 3 months, but in some cases it does not. And with the time, after 3 months, you wait. With the time, it might heal, but it might also not heal and become a non-union, which is the stage where the two pieces of bone will never heal. So imagine you end up with a tibia in two pieces that will never heal. So then it requires very heavy surgeries, rescue surgeries, in most cases, several rescue surgeries to finally complete the fusion. So what we do is we intervene earlier, and we inject our cells in those patients that have difficulties healing, and we inject those cells so they regenerate the bone. They do two things. They will build bone by themselves, our cells being differentiated, and they will also call on the own cells of the patient. So we completed the phase 2A trial last year. I will share some of the results with you, and we're preparing the next phase, which would be the last phase of clinical development to be started before the end of the year. And then finally, our non-cellular product, JTA004, it's an improved viscosupplement for osteoarthritis of the knee. In that case, an extremely large indication. About 27 million patients suffer from symptomatic osteoarthritis of the knee in the large Western markets. We completed a phase 2B and are moving into a phase 3. The objective here is to put on the market a product that has better pain-relieving properties than what is currently on the market, and I'll show you that later too. So if we talk about ALLOB, or allogeneic bone cell therapy platform, it's pretty simple actually. We're using a bone marrow from a healthy donor. We isolate the mesenchymal stem cells present in the bone marrow of this healthy donor. We amplify those cells, and we differentiate them into bone-forming cells. So those cells have bone-forming properties when the product is finalized. The final product is also cryopreserved, which allows a very easy supply chain, storage, transport. And then this product can be easily administered in a minimally invasive way, locally, for example, in the fracture that needs to regenerate better. Out of one bone marrow donation, we're currently able to produce about 100,000 doses for 100,000 patients, which obviously has large advantages and puts us in a position where this product can be commercialized. So if we're talking about delayed union fractures, as I said, we're talking here about those severe fractures, for example, after a car accident, a sports accident. Those fractures normally heal in 3 months, but in some cases, in particular in patients that have risk factors like open fractures, high energy fractures, smokers, etc., in some patients it's much more difficult to heal. So those patients are left with nothing 3 months after the fracture. If it has not healed, well, there is nothing offered to them besides wait and see, and that's where we intervene. We inject the cells in the fracture to avoid that these fractures finally turn sour and become non-union, which will lead to a lot of rescue surgeries. Recently, I met a patient that had had 90 rescue surgeries, so you can imagine what the life of such a patient becomes after a very simple tibia fracture in a car accident. So we're trying to avoid that and inject the cells right in the fracture interline in a very minimally invasive way. So you have here an image of what this looks like. So the fracture on the left was about 6 months old. You don't need to be an expert radiologist to see that there is obviously a problem with that fracture. The surgeon came and injected the cells right in the fracture interline. After 6 months, the fracture was finally consolidated. We had a phase 2A trial with 21 patients followed up for 6 months, where we injected those cells. Those fractures were on average 6.5 months old, so rather old fractures. Almost nothing had happened to those fractures in the previous 6.5 months. After we inject, we see that on average, our patients improved by almost 4 points on the tomographic union score, which is a scale that is used to just evaluate the progression of the fracture. It was predefined that the required minimum increase to be considered a responder was 2 points. On average, they improved by 4 points, so almost double what was expected. So we saw here good progression of the fracture. We also have clinical endpoints in terms of general health status and pain, which also progressed well in those patients that had lived with such a fracture for more than 6 months. If we're looking at JTA004, so just to move back to this one. So this one has completed the phase 2A, where we will be starting the last phase of clinical development in Europe and in the US, and submit that to authorities before the end of the year. If we're looking at JTA004, it's a non-cell product. But but even if it's non-cell, it might have an interest as well. I know we're all focused on cell and gene here. But this is, we're here talking about osteoarthritis of the knee. Very prevalent condition, increasing pool of patients because the risk factors are age and weight, and both of them are increasing, both the age and the weight of the population. There are a series of treatments given to those patients suffering from osteoarthritis of the knee. So it's a degeneration of the cartilage that leads to a lot of pain and difficulties functioning. So a series of treatments are offered. At some point, they get a viscosupplement prescribed, which is an intra-articular injection of typically hyaluronic acid in the knee to reduce pain. This is a very big market. It's a $2 billion market worldwide, but still those products don't have a lot of efficacy. Some patients will be good with them, some patients not at all, but on average, the studies have shown that the results are not fantastic. So we are developing an improved viscosupplement that contains hyaluronic acid, like other viscosupplements, but also 2 additional active substances, one analgesic product and one enriched protein solution. And the combination of those 3 elements forms JTA004, which we hope will show better pain relief in those patients suffering from osteoarthritis of the knee. We did a phase 2 trial with 164 patients with a 6 months follow-up. In those trials, it's a very classical type of design. We look at the pain relief, the pain reduction between baseline and 6 months. And in this trial, we compared our product JTA004 to Synvisc-1. Synvisc-1 is the market leader. It's a product by Sanofi. It's a specific type of hyaluronic acid. They sell for between 300 and 400 million euros a year. But again, not always with the best results. And this is the results some of the results we found. We looked at the pain reduction, and on the light blue bar is Synvisc-1. We saw a pain reduction of between 15 and 16 millimeters. Pain is measured in millimeters on the Womack pain subscale, while the JTA group showed a much better pain reduction, around 26 to 27 millimeter, both at 3 months and at 6 months. So these results are very encouraging because they show that we may have something here that provides much better pain relief and therefore will delay as much as possible total knee replacement longer. Therefore, we're moving into a phase 3 trial in Europe and in the US that we anticipate to submit to the authorities before the end of the year as well. So that's the second phase 3 trial that we will be submitting to authorities before the end of the year. So if we're looking at what we're doing, we're offering off-the-shelf solutions, and I don't have to tell you that for cell therapy products, off-the-shelf, easy-to-handle products are a must when you want to do more than clinical trials and you want to commercialize a product. So both with the allogenic product that has a high yield and is cryopreserved, and obviously the viscosupplement, we have off-the-shelf, easy-to-handle solutions targeting large and underserved populations. Again, very little innovation in orthopedics. We have accumulated more and more clinical data. We will accumulate more this year in the summer when we will have the final results of our spinal fusion, lumbar spinal fusion trial, phase 2A trial. So we have accumulated a lot of clinical data giving us confidence that these products can bring a lot of value to both surgeons and patients. And finally, we believe that we have products that are commercial proof, that can be scaled up easily with production processes that are as simple as they can get, certainly for cell therapy. In terms of partnerships, we're looking for partners to either co-develop or commercialize the product in various parts of the world, and I'd be very happy to discuss that with those of you interested in those type of collaborations. And I think I still have 2 minutes for questions if any of you has questions.
What's the allogenicity of the cells?
You mean in terms of immunogenicity? Yeah. So obviously this is the big question when you work with an allogenic product. This was one of the things we're measuring in the phase 2A trials. What we've seen very good tolerability of the product. So we haven't seen anything worrying in terms of immunogenicity. So that's one from a clinical perspective. Then we're looking at blood samples of those patients receiving the product. So there is no matching needed between the donor and the recipient, the patient. What we're doing is collect blood samples. What we've seen is that in about half of the blood samples, there were no donor-specific antibodies that we found. So those patients didn't realize at all that cells from someone else were injected in their body. In the other half, there were donor-specific antibodies that were either pre-existing after pregnancy or transfusion or something, or developed after the administration, but again, without clinical consequence. When you look at clinical phase 3 clinical data of other allogenic derivatives of mesenchymal stem cells that were published, you see the same type of percentages. And I'm thinking here of a product that just got marketing authorization in Europe. So we're in the same ballpark.
So you see this as integrate or just promoting the natural endogenous regeneration?
So what we see our cells do is two things. First, there is a clear osteogenic potential, so they will create bone by themselves. We see that in our mice models, where we see human bone being created in the mice. That's one part, but we also see osteoinduction, so the fact that those cells will release factors that will call on the own cells of the patient. So we also see that because in our animal models, we see mouse bone being created. Those two types of bone will integrate. With time, the human bone will disappear because this is obviously a naturally regenerating process. But in the beginning, there is really the dual effect in terms of mechanism of action.